nucleotide here. So we have a phosphate group, 5-carbon sugar, and we have a nitrogenous base, many nitrogen atoms in the ring instead of carbon atoms. These three structures bonded together is a nucleotide, just like a bead on a necklace. Okay? And we have 2 chains of these nucleotides that are then hydrogen bonded together and twisted around each other. That's DNA. We covered that last class. What I want to point out to you with this particular slide is the sugar. I had a question on this, I think, in recitation. Right? Here's the sugar. The sugar has a hydroxyl group right here on the 3rd carbon. And on the 2nd carbon, there is not a hydroxyl group. There's just a hydrogen atom attached to that carbon. This is a way for you to distinguish between the 2 types of monosaccharides, the 2 simple sugars, deoxyribose and ribose. Deoxyribose does not have a oxygen here at the 2nd carbon. So we say it's deoxy. It's without oxygen. So this is deoxyribose. Therefore, if this sugar is deoxyribose, then this nucleotide, this whole group of 3 substances bonded together, is from DNA. This is a DNA nucleotide. Just knowing that this sugar is deoxyribose. It's not important for me, at least for now, that you recognize the different nitrogenous bases and can tell them apart, although you probably will be able to later on. Just so that you know that this is a nitrogenous base, then that's fine. Okay, I'm not going to expect you to tell from the nitrogenous base structure if it's a nucleotide from DNA or if it's a nucleotide from RNA. From the sugar, yes, you should be able to tell. You look for the 2nd carbon, and off of that 2nd carbon, if there's no oxygen there, then the sugar is deoxyribose. So the nucleotide is from DNA. Does this make sense? Yes?
So in RNA, there will be oxygen, so it's going to be OH and another OH?
Yep, I'll show you that right now. Okay. Okay, so here is an RNA nucleotide. If you look at these slides quick, you might think, oh, she put the same slide in by mistake. Right? I didn't. The title's different, right? This is an RNA nucleotide. If we were looking at RNA off of the 2nd carbon, now there is a hydroxyl group there. There is an oxygen and a hydrogen there. That makes the sugar different. This monosaccharide is not deoxyribose. It's called ribose. So ribose has an oxygen at the 2nd carbon. Deoxyribose does not. And you can tell just from looking at the nucleotide, specifically looking at the sugar, that this is ribose and this is a nucleotide of RNA. This part, the nitrogenous base that's in blue here, that's going to change. That will change depending on if we are looking at an adenine nucleotide or guanine nucleotide or cytosine nucleotide, right? So this part will change. The nucleotide doesn't always have to look exactly like this. This will stay the same. This will stay the same, at least in RNA, and this part's going to change. You might see this nitrogenous base have either 1 ring, like you see now, or it might have 2 rings. I was hinting to this last time. I'll flip back again to the DNA nucleotide so you can see what I mean, right? You see here in this DNA one, there's 2 rings. There's 2 rings in the base. You could have a 1-ring base here too. The only thing that is different between the 2, at least for your purposes so far, is the sugar. The sugar is what makes it DNA, or the sugar is what makes it RNA. So if we look at these bases now, yes, there's a 2-ring base and there is a 1-ring base, and we give them names. We say that, you know, this particular base, because it has 2 rings, is called a purine base. You'll see that here. We're just looking at the base, just looking at the blue structure, 2 rings bonded together. This one here, since it's just 1 ring, is called a pyrimidine base. The significance of this is when we hydrogen bond. We were looking at 2 strands of nucleotides and, you know, the sugar phosphate backbone on the sides, and then we talked about the hydrogen bonding between the bases. The hydrogen bonds will only form between a purine base and a pyrimidine base. You'll only have a hydrogen bond between a base that has 2 rings and a base that has 1 ring. You won't have a purine hydrogen bonded to a purine. Are you with me? You won't have a pyrimidine hydrogen bonded to a pyrimidine. It's only purine hydrogen bonded to pyrimidine. So when I said last time that I like to look at George Clooney, right? Okay, it's that way, right? A hydrogen bonds to T because one of them is a purine base and the other is a pyrimidine base. And same thing with George Clooney, right? The G is a purine base and the C is a pyrimidine base.
Ms. Brown?
Yeah.
For that slide,
Backwards, yeah.
mine has uracils.
Okay, so
Not-
so like with the other slide that I said you might think I put it in twice and made a mistake. I didn't. I showed you the nucleotide in DNA and in RNA, and then I showed you the bases in DNA and in RNA. It's okay because I've already had a question on that, so that's why I'm you know, doing this. So that's fine. So with this slide here, where you can see guanine, adenine, cytosine, and thymine are the 4 bases that I'm referring to here, I'm talking about DNA. Because thymine in particular is the pyrimidine base that's only found in DNA. And if you could recognize that this is thymine, I don't expect that you do that, but if you could, that would be another way for you to identify DNA. If I just showed you a nucleotide like this, and you maybe forgot to look at the second carbon and see that it's a deoxy here, you could recognize this as thymine. But I don't expect you to do that. Thymine is only found in DNA. The adenine, the guanine, the cytosine, they can be found in either DNA or RNA. So the base structure is not always going to help you. Look for the sugar.
So the adenine can bond with thymine, but it can't bind with the cytosine?
Correct.
So it's just like, it's very specific.
It is.
So thymine is replaced with uracil
Yes.
in
When-
RNA?
Yep. And if you flip to a few slides from there, you can see, you know, looks like the same slide, but it's really not. This time I have uracil here, because here I'm referring to the RNA nucleotides and the bases specifically found in those RNA nucleotides. You should notice that the deoxyribose is no longer there, that now we have ribose as our sugar, and this base could be uracil. It's not going to be thymine. There's no T in RNA. Wherever there would be a T, there's a U in RNA. No T in RNA. No. So, so if I have a sequence, A, T, T, A, G, something like that. If I give you this sequence, For each of the letters that I wrote down, the letter represents the base. The letter represents this part in blue. Attached to the blue base is the sugar. And in this case, what would the sugar be? Deoxyribose. And then attached to the deoxyribose would be a phosphate group. I'm not drawing that all, but you assume that that's there. Now, how did you know it was the sugar deoxyribose?
T.
Good, because I put thymines here in my sequence. I have T's, which is for thymine. If you see T's, you know it's DNA. If I wrote another sequence and maybe it is A, U, G, C, A, U, something like that, then from me writing the U, you know that that's an RNA sequence. With me? Yes? So what would the sugar be in that sequence? Ribose. And even though I'm not drawing it, there's a ribose and a phosphate group attached to each one of those letters. So the structure of the nucleotide is basically the same. We have the phosphate group, the sugar, and the base. The base could be different. It could be adenine, it could be guanine, it could be cytosine, it could be thymine, or it could be uracil. But always when we hydrogen bond together, there's a pyrimidine and a purine hydrogen bonded. Specifically, the A bonds with the T and the G bonds with the C. The bonding doesn't happen in any other way.
So does the A bond with G then?
So when we are forming RNA, when we're making a protein and we're going through the process of transcription, the A is going to correlate to a U. And you'll see this in chapter 3. We'll take it another step further in chapter 3.
So once again, purines will only go bond with purine.
Pyrimidine.
Purine.
Yep. Right, so these are the 4 nucleotides in DNA, where I was just writing the letter A, T, T, A, G, C. The whole nucleotides would look like this. If this is the A that I'm writing, I mean all of this as the A. If this is the T that I'm writing, I mean all of this as the T. And you can look over here at the second carbon, at the sugar, and you'll know immediately that it's deoxyribose. If you can't recognize the T, the thymine base, from the cytosine and so on. You should at least know which ones are purine bases and which are pyrimidine bases. The purine bases have 2 rings, and they're the adenine and the guanine. Those are purines. The cytosine, the thymine, those are pyrimidines. They have 1 ring. So without showing you uracil, what would you expect uracil to be? Would uracil have 1 ring or 2 rings?
1
It would have 1. How do you know?
takes place for T.
Right, because the adenine has 2. It's a purine, and it has to bond with the pyrimidine, so we know that that uracil is going to have 1 ring. You always have a 2 ring, hydrogen bonded to a 1 ring. Athena, yeah? I might show you a picture. I think I have one. Let me end it. Something like this. I might show you a picture like this. And have you highlight the nucleotide. So if you're going to highlight the nucleotide here, let's say is 1 nucleotide, right? Okay? I could ask you, is this base right here a purine base or a pyrimidine base? It's a pyrimidine base. I could say, is this sugar ribose or deoxyribose? deoxyribose. I don't have to tell you the letters. I don't have to tell you that this might be, a T, for example, or a C. I don't have to tell you that. From what you know so far, single base, single ring here is a pyrimidine. And it's going to bond to a purine base. So if this is a purine base, what are the possibilities? It's either what?
Adenine.
It's either an adenine or a
Guanine.
guanine. It's either an adenine or guanine. So I might show you a picture like this one and have you identify the nucleotide or the sugar phosphate backbone or the base pair. Right? Where would the base pair be? Is this the base pair?
No.
No. This is the base pair, right? Yes? This base, nitrogenous base, bonded to this one. Through hydrogen bonds, that's a base pair. Here's another base pair. Now this is DNA. Two chains or strands of nucleotides, then hydrogen bonded together and coiled around each other. If we were looking at RNA, it's just one single strand. It's just one strand. And we will use one of these strands of the DNA to make the RNA. We use the DNA as a template to make the RNA. So if I told you, I think I erased my writing before, but that's okay. If I said that I have an A and a T and a G in my DNA, and I wanted to make RNA, then all I have to think about is the base pairs in RNA. Remember, there's no T in RNA. So instead of the A pairing with T, if we wanted to go to RNA, A is going to pair with a U, right? And so over here, I would have a U. Now, what's the pair for T? A, then I would have an A here. And what's the pair for G? A C. So I would have UAC as my 3 nucleotides in the RNA. Yeah, with me?
Mm-hmm.
Practice with that, because you absolutely have to remember the I like George Clooney. The I like to look at George Clooney. ATGC. You have to remember it. If you don't remember it when we get to protein synthesis, it's going to be even harder.
So the structure of the DNA is pretty much the sugar backbone and
I definitely, I will ask you the sugar phosphate backbone. I could ask you, what type of sugar it is. Is it a purine base or a pyridine base? Where's the base pair?
And the base pair is what, in that picture, the little H's, that's the hydrogen.
I think they were blue. The 2 blue nitrogenous bases, hydrogen bonded together.
And that's what
That's the base pair.
shape, right?
Well, I mean, it's the interaction between the hydrogen bond and everything else that basically makes it. When we go from DNA to RNA, it's going to uncoil as the process happens. Questions on this? Any of this? We did talk about ATP a little bit, I think, too, last time. And is ATP a nucleic acid?
No.
No. Why?
Energy.
Okay, it's energy. It's like money. Does it carry genetic information?
No.
No, it doesn't. Okay. It might be similar to a nucleotide because it has a sugar, it has phosphate, and it has a base. But the base is always adenine, right? It's always that purine base adenine. And there's 3 phosphate groups in ATP, adenosine triphosphate, tri-3. And when we're using this energy, we can break these bonds between the phosphate groups and release the energy. Then we would go from ATP to ADP. If I remove one of these phosphates from ATP, where it's triphosphate, if I take one phosphate away, then what do I have? Which is, what's the D? Di. Means 2. Right? And I can even take another phosphate away, and then I would have AMP, monophosphate, adenosine monophosphate. Okay? So the energy is in between these phosphate bonds. There's no genetic material here. And we will definitely come back to this more too. All right, so let's look at chapter 3. See where DNA is over here? This is in chapter 3. I just skipped ahead a little bit. You can see the DNA molecule, which is the double helix up here, and how it's opening up. It's essentially separating the two strands by breaking the hydrogen bonds, so that we are going to either replicate this piece of DNA, make a whole nother exact copy of it, or we can use one piece, one strand of this DNA as a template to make the RNA. So we're gonna come back to that a little bit later. Today we're gonna focus mostly on the structure of the cell and on movement through the cell membrane. And I've already given you some of the lab supplies because, you know, as we talk about the lecture, I'm gonna have you start to do part of the lab. So it will take a little bit of time for us to see some of the results. Okay, so we're gonna be looking at cells. This should be review for everybody. From basic high school biology, or Bio 105 if you took that. I think most people will recognize that this is a cell, and Lord, I hope that you do. At this point, this is an animal cell. What's the outer boundary of the animal cell called? Plasma membrane or cell membrane. We talked about this last time. This was the phospholipid bilayer that I was referring to, the 2 layers of phospholipids, or the peanut butter sandwich that I said. Okay, so here's the phospholipid bilayer, this nonpolar covering all the way around the cell. It's a fluid covering because the phospholipids can move within the bilayer. There can be proteins embedded within the phospholipid bilayer. I showed you this last time as well. And it will regulate what comes in and out of the cell. It determines what's going to allow to enter and what it's going to allow to leave. Most people will know this center region here that's in purple. So what's that? It's the nucleus. That's where the DNA is. This is also where the RNA is located that we were just talking about. So the DNA is here within the chromatin or chromosomes. The RNA is right in the center. Here in this darker area, it's called the nucleolus. It's the cluster of RNA. So I might see my uracil bases only over here, but some thymine bases will be here. Okay. We will look at the cytoplasm. That's all this light blue in the background. And we'll talk about the cytoplasmic organelles. In the lab packet that I gave you, that's the yellow packet, you have a picture of this model. I tried to make it as big as I could. Should be kind of in the middle-ish. Okay. So if you wanted to, you know, label it as we talk about it, you certainly can do that. But you're looking at an animal cell. We're not going to look at plant cells. This isn't a botany course. In Bio 105, we would have talked about plants and we would have looked at bacteria as well. We're just going to focus on the animal cell. So light blue in the background, that's the cytoplasm. Sometimes we'll refer to it as the cytosol if we're just looking at the liquid component. This outer boundary is the plasma membrane. We looked at the nucleus. And the nucleolus in the center of the nucleus. Yep, the nucleus is in purple. It's right here. It's this whole structure here. It's all the nucleus. And in the middle of the nucleus, a smaller circle is usually a darker dot in the middle of the nucleus. That's the nucleolus where the RNA is. So from previous knowledge, The colored structures within this model, like the rough endoplasmic reticulum that you can see here, it's close to the nucleus, and the Golgi body or Golgi apparatus, it's this structure that's in red. Both of these structures are going to be very important when we talk about protein synthesis. And we'll be looking at the DNA converting into RNA in the nucleus, and the RNA leaving the nucleus and going to the ribosomes on the rough ER to make a protein. And the protein that we're making by bonding the amino acids together through peptide bonds, through dehydration synthesis, that protein that we're making here at the ribosomes is then going to go to the Golgi and be modified and changed, perfected. We might start with our primary structure over here, and then over here we might be adding some tertiary structure and so on. At this point, when it's leaving the Golgi, it might go out of the cell. You know, if it's a hormone that we're making within the cell, if the hormone is our protein, it might leave the cell and go to its target. Or it could be something that the cell needs, and it might stay inside the cell. It could be a protein that's needed intracellular. These are going to be the most important ones that we will focus most of our attention on when we get to protein synthesis. The rough ER, which is here with the ribosomes on it, and the Golgi, that's over here. So you'll see some dots scattered about. There's some red dots. I don't know if you guys can see the color very well, but the red dots, and then there are some blue dots. These are the secretory vesicles. Red ones and blue ones. The color will actually tell you where the vesicle came from. A vesicle is like a bubble. You know, if you blow a bubble, it's going to float, it's going to move. Right? But inside of these vesicles are products, are proteins that were just made. The blue ones, for example, contain proteins that were made at the rough ER, and they're going to the Golgi. They're using the vesicle as a transport mechanism. The red vesicles contain a protein that was modified at the Golgi and is leaving the Golgi. The color tells you where they came from. Blue came from rough ER, red came from Golgi.
And they're called what again?
They're called secretory vesicles. It's kind of like the, you know, the good witch Glinda travels in the bubble. All right, these orange structures, hopefully you'll remember as well. Does anyone know? What are these orange ones? Good, good, good. These are mitochondria. And mitochondria are for ATP production. It's the orange right here. For the production of ATP. I call it the powerhouse of the cell.
That's for cellular respiration.
Yes, cellular respiration. Aerobic cellular respiration occurs here at the mitochondria, where we're making the ATP. We have the adenine, and we're going to bond to it the ribose, and then the 3 phosphate groups.
And they also use secretory vesicles to get the nucleic acids.
Mitochondria?
Yeah.
No. Nope. Mitochondria are unique because they actually have their own DNA. Mitochondrial DNA. We'll talk more about this with the reproductive system, but all of the mitochondria that you have within your body came from your mom. Right? None of them came from your dad. Mm-hmm. Yes, you can thank your mom for all your energy. Right? Yeah. When the sperm fertilizes the egg, the only thing that is deposited into the egg, essentially, is the paternal DNA. It's just the chromosomes from dad, nothing else from the cell. All the components of the cell, specifically the mitochondria, are there already within the egg. So all your mitochondria, you trace them back in your maternal line. So they have their own unique DNA, and there's this theory that the mitochondria themselves were actually once bacteria that survived on their own, single-celled organisms, and then moved into another cell and said, yeah, I kind of like it here, I'm going to stay here, and we're going to live in symbiosis together, this cell and me. They look like bacteria. They look like little capsule-shaped bacteria. Same with the chloroplast within plant cells. That's pretty much it for now. We'll look at peroxisomes and lysosomes a little bit later on. That's the white circles and the yellow circles that you see. Peroxisomes and the lysosomes, they're basically for digestion. Right? The lysosome is like the cleanup crew. It's like the garbage. If we want to get rid of something or destroy it, we'll put it in the lysosome. Those are in yellow. One of your white blood cells, a macrophage, for example, might eat or ingest a bacteria that it knows is foreign, and it will destroy that bacteria by merging it with its lysosome. So it's kind of like the stomach on the inside of the cell. Right? Here's the bacteria, here's the lysosome, you merge the two together, so now they're in one membrane. All the acids and everything that was in the lysosome is now, digesting the bacteria cell. So the lysosome has a membrane around it, just like all of the vesicles do, the blue and the red vesicles we were looking at before. The rough ER and the Golgi also have a membrane around them. And when I say membrane, I mean just like this one, the phospholipid bilayer. Right? 2 layers of phospholipids, that's considered a membrane. And all of these organelles, rough ER, Golgi, secretory vesicles, lysosome, they're all membrane-bound organelles. They have a phospholipid bilayer around them. If the lysosome did not have a membrane around it, what would happen to the cell? It would digest itself. Yeah, a programmed cell death, apoptosis, is essentially, the lysosome bursts, let's say, and the cell dies because of that. So we'll look at the parts of the cell. Did I miss anything, or is everybody okay with this model so far? You got enough lines on your paper? Yes, smooth ER. Sure, the smooth ER is right here. Smooth ER is not involved in protein synthesis. It is involved in lipid synthesis, though. But smooth ER is here. Smooth ER is considered smooth because there's no ribosomes attached to it. And if there's no ribosomes, then it can't make proteins. And this part right here in the middle, these are called centrioles. We'll look at these a little bit later. These help to move the chromosomes around when the cell's dividing through mitosis and meiosis. They're called centrioles. But first, we're really going to focus on the plasma membrane of the cell. Right? I just showed you a typical animal cell. Typical. Doesn't mean every single cell in your body looks exactly like this. Okay? If I was to look at a red blood cell, it wouldn't look exactly like this. Red blood cells lose the nucleus. This part where the DNA is, it kicks it out. As a red blood cell matures, it ejects its nucleus. It says, I don't need these instructions. And it kicks them out. Why? Because it has millions of molecules of hemoglobin protein in the cytoplasm. And what's the hemoglobin for? Binding to oxygen. And if the nucleus is there in the cytoplasm, then that means there's going to be less hemoglobin able to fit there and less oxygen able to fit there. So it kicks out the nucleus. Here's a red blood cell right here. And you can see that it has a dip in the middle of it to show that there's no nucleus there. We'll look at this under the microscope today. We'll look at all of these actually under the microscope today. If there's no nucleus in a red blood cell, can it make proteins? Nope. Doesn't have any instructions to do that. What if it gets, what if an organelle gets damaged? Can it fix it? No. Doesn't have any instructions. Okay, so they only last for, certain length of time, certain number of days because of that. They have no ability to repair. They don't have any DNA, any genetic material. This is a nerve cell. It's a neuron. I have a model of this. I'll show you this under the microscope too. It looks nothing like the typical cell. Nothing at all. But the structure helps with its function. Just like you saw with the red blood cell, where it loses its nucleus so it has more room to carry oxygen, the nerve cell is shaped like this because its major function is communication. Transferring messages, sending information from maybe your fingertips to your brain and processing that information. Sending a response from your brain out to your smooth muscle in your skin to cause your hairs to stand up when you get cold. So the neuron, this nerve cell, is very long because of that. And this break here, people get confused by this. This break just means that this neuron is so long they can't show you the whole thing. So they're just going to show you the beginning and the end of it. That's what this white area means here. You can see the nucleus though in the neuron. Here's the nucleus here. And this little dot inside the nucleus, what's that? That's the nucleolus. So is this neuron capable of making proteins, you think?
Yes.
Probably. And if you look closely, you might see some of the raphiar around the nucleus, and you can see some mitochondria here as well. So the goal for lab today is to look at cells and be able to identify the organelles, the parts, to look at them not only in the models but also under the microscope and be able to identify the parts. And then we'll talk about how materials can move through the cell membrane, through the phospholipid bilayer and the plasma membrane. I did not show you flagella. This animal cell does not have a flagella on it. What animal cell has a flagella? Okay, but animal cell, I said.
Sperm cell.
Sperm. Sperm's really the only animal cell that has flagella. Bacteria is not an animal cell, but it is a prokaryotic cell. And you're right, bacteria can have flagella, but they're prokaryotic. This picture here, is not totally accurate. You really will not have all of these structures in one cell. You really won't have microvilli and a cilia and a flagella on the same cell. It doesn't really happen, okay? We might have microvilli on one cell, we might have some cilia on another one, we might have a flagella on another cell. They're just trying to show you the comparison between the size of each of them. Microvilli being extensions off of the cell that are very tiny, micro, like you need a microscope to see them. Cilia are a little bit longer and mostly there for movement. You have cilia lining your respiratory tract. So you have mucus within that lining and you're moving the mucus down your throat, things like that. Cilia are going to help with that. You have cilia lining your, well, I shouldn't say your because not everyone's female, but fallopian tubes. It's the movement of the cilia that help to move the egg from one end of the fallopian tubes towards the uterus. The flagella is obviously the longest of all of them. And it is for movement too, but movement of the whole cell, movement of the whole sperm. Whereas the cilia are just kind of moving what's on top of the cell, not what's in the cell. All three of these, the microvilli, the cilia, the flagella, are all part of the cytoskeleton of the cell. Cytoskeleton is this framework. You can see these kind of tubes or bars on the inside of the cell that hold all the organelles in place. It's like a scaffolding within the cell. It provides the shape for the cell, and it can help to drive movement of the cell, parts of the cell and the whole cell. part of the cytoskeleton. So one cell's not going to have all of these. I showed you this picture last time, I think. All right, so here's the phospholipid bilayer. The light blue circles are the hydrophilic heads, and the gray kind of lines, those are the hydrophobic tails. One phospholipid has one head and how many tails? Two. Good. Okay. 2 tails from each of these heads. And you can see embedded within the phospholipid bilayer are some steroids. This is cholesterol embedded within the bilayer. Cholesterol is on the side of the phospholipid tails. So is cholesterol polar or nonpolar? Good, it's nonpolar. It's lipid, right? It's fat, just like the hydrophobic tails, like with like. The cholesterol will orientate itself in the tail portion of the membrane. And we have tail to tail arrangement. This whole thing is the phospholipid bilayer. When I said membrane-bound organelle, like the membrane around the lysosome or the secretory vesicle, I mean this whole arrangement. It might not have the same exact proteins as the outer boundary of the cell plasmamembrane has, but it's the same arrangement of phospholipids, tail to tail. So look at some of the proteins that are embedded in the membrane. We talked about the structure of proteins last time. Proteins are made, proteins are polymers of what monomer? Good, amino acids. Amino acids are the building block. And I would take an amino acid and peptide bond it to another, and keep making this long chain. And then I might hydrogen bond them even further and coil it or fold it. And what I'm left with is something like this, the protein. You can't see all the individual amino acids in there, but the overall shape of them, you can see. And this protein might, serve as an attachment from this cell to another cell. Kind of like a staple, stapling the papers together. Protein's gonna attach the 2 cells together. Or this protein over here might have a certain pleat or a certain helix arrangement to it, so that the middle of it is like a pore, it's like a channel. And it has a plug at one end. It's like a door. And this plug has the ability to move out of the way and open up the channel, or to come back and close the channel again. This protein can serve as a gated channel. And only when we reach a certain charge on the membrane will this gate open. Only if we reach negative 55 millivolts will this gate open and ions can come into the cell. Or if we reach positive 30, the gate might close. You know what I mean? Yes? We have some condition that's going to open and or close this gate. So the cell membrane's regulating what's entering and what's leaving the cell. The protein is doing that. Same thing over here. Another protein on the inside of the cell membrane, and it might have some carbohydrates attached to it. All these purple structures are carbohydrates, right? They look like glucose, don't they? Like glucose bonded together. But now the glucose chains are bonded to a protein. So we have a carbohydrate and a protein together. We call it a glycoprotein. You see the term right here. Right? It's a protein, which is the orange part, but it has a carbohydrate part to it. And that carbohydrate part might serve as a receptor. Maybe a hormone coming along can bind to that receptor and elicit some change. You got a question?
This might be too literal, but would salmon be a protein?
Salmon itself, like a fish?
Yeah.
Okay, yeah. So the organism, the salmon, the muscle of the salmon contains proteins. Just like chicken, the animal, contain, the meat, what you would think of as the meat of the chicken, contains proteins. It's not in itself a whole protein, but in the muscle there's actin, and myosin proteins, so that's your source of protein.
So when you're ingesting, that's your body's getting.
Yep, that's how your body's. It's basically you're eating the muscle of the animal, and you're breaking the muscle down because the proteins are within the muscle. It's the same thing as if you were to eat any animal. You know, people don't eat dogs typically or cats, but it's the same muscle, it's the same actin and myosin.
So the carbohydrate on the glycoprotein
Yep.
is not being absorbed in the cell though? It's
Correct.
just like hanging out
Yep.
on top of the
Mm-hmm.
membrane?
Yeah, it's just sitting here, and that's its job, to just stay there. It's not moving in or out of the cell, it's just there, it's just staying there. And its job is to serve as maybe a binding site for a hormone to bind to. Or its job might be to say, okay, you know, I am a cell from Melissa's body, and all the white blood cells that are coming by and scanning to make sure, you know, I'm healthy and everything. I know that those cells belong to me because I have this little, you know, flag or something on the surface. You know, you're like the Yankees, so you hang a Yankee flag outside your house. It says you're a Yankee fan. Or a Red Sox, or I won't judge you then. But, you know, so this could be one of those like identification proteins that says, I belong to this person. And if another cell is there and it doesn't have that flag, then your body, your immune system knows to target it and destroy it because it's invading your body. So proteins have many different functions within the cell membrane. And I said before that this cell membrane is fluid. Pretty sure I said that, right? The phospholipids can move. You can have proteins move within the phospholipids. Like this protein here is not totally stuck here. It's not glued in position. It can move over here, or it can move back. It's fluid. It's like a layer of bubbles. And the proteins can move across within those bubbles, and they're not going to pop. They're not going to pop. We say this is the fluid mosaic model. Right, it's a fluid mosaic model. That model of the plasma membrane. Why is it fluid? What can move? Okay, the phospholipids can move. The proteins is what you were going to say. The proteins can move within the membrane. Fluid mosaic model. Why is it mosaic?
Feels like a
Probably a better term than maze.
All the organelles.
Not necessarily organelles, but you're on the right track. If I walk into a mansion, I say, oh my God, this is a beautiful mosaic tile floor. What am I talking about? What's the mosaic? Is it just one tile, straight, plain, boring like that? It's like a pattern. It's like a pattern. It's all different tiles, maybe different colors, maybe different shapes, making a pattern on the floor. So that's why we say mosaic, because there's all different proteins with different shapes and different arrangements that make up this plasma membrane. And the plasma membrane from one cell is different than the plasma membrane from another cell. If this cell is going to be a target for a hormone, it might have a protein on it with a receptor for that hormone. Whereas another cell would not have that protein, it would not have that receptor. So not every cell is exactly the same, where it's going to have exactly the same proteins and exactly the same receptors and flags and all of that.
How do enzymes also bind certain proteins?
Yeah, you can have, I don't know if I'm happy to do it. So you can have a protein that's embedded within the phospholipid bilayer, like they're showing you here. And that protein in itself is an enzyme. So where I was drawing that weird shape last time with the active sites in it, that protein is now embedded within the phospholipid bilayer. And if A and B want to come together to make C, they can do that right at the surface of the cell, because the cell has this enzyme, this special protein to do that. The proteins within the cell membrane can have many different functions. I think I've hit on pretty much all of them. You do not really need to identify them. I will not show you a picture and say, is this a cell marker or is this an enzyme? But you should have a good idea of the different types of proteins that you might see in the cell membrane. Questions on any of that? No. Okay. I would like at least one person from each group to grab their beaker of nothing right now and fill it halfway full with water, if you wouldn't mind. There's the sinks in the back of the room. Should take maybe 1 second for you to do that. Doesn't have to be exact, just rough, half full of water. Yeah, and then a substance like sodium ion maybe would move from the outer portion of the cell into the inside of the cytosol of the cell. Doesn't have to be exact. Should take you like literally 1 second. Done.
I saw it on
Right, there's just water in your beaker, right?
active.
Only water? You just put water in there? Half full. Okay, that's
I
not
see
quite
them
half
online.
full.
They have like different.
That's fine,
colors.
that's fine.
Online.
Okay,
Okay.
carefully take either, it doesn't matter, a drop of IKI or a drop of the methyl blue at your desk and drop a drop of it into the water in the beaker. Be careful, don't get it all over your skin and your clothes. They are stained, they're dyes, so just like food coloring, you know. One drop is fine, not the whole bottle. It's all right. Did everyone put 1 drop in? Either one, don't matter. You know, some groups did the blue, some groups did the iodine. Either way is fine. Okay, so we're gonna look at how materials can move in and out of the membrane. Okay, the plasma membrane of the cell is selectively permeable. That means it's going to decide what it's going to allow to come in and what it's not going to allow to come in. Just like you, when you answer your door, you can decide to answer your door and let the person in, or pretend you're not home and not answer the door. If the substance is able to easily go into the cell, pass into the cell, we say that that type of movement is a passive movement. It does not require energy, it does not require ATP in order for the substance to move from the outside of the cell to the inside of the cell. It's passive. If something requires energy, it's obviously not passive, but it's active transport. We'll focus today on the passive. In lab, we're going to specifically focus on the passive. Movement like diffusion, which you're testing now, by the way, and osmosis and facilitated diffusion. All of these are passive types of transport. You do not need ATP to move substances in this manner. You don't need any energy. Yeah? Centrioles? Yes, because you're basically moving chromosomes around within the cell, and any type of movement of the cell and the membrane involves active. Okay, so with diffusion, osmosis, and facilitated diffusion, we are moving a molecule or molecules from an area of high concentration to an area of low concentration. Okay, what did you just do? You took the blue or the iodine and you dropped it into a beaker of water. You had in your beaker of water nothing, just water. no iodine and no blue to begin with. So your beaker was the low concentration, it was 0. And you've added a drop of methylene blue dye or of IKI. The area where you dropped the dye in is now darker than the other areas, yes? Yes? So the area that you dropped the dye into the beaker has a higher concentration of dye molecules than the rest of the beaker. And if you watch it over time, and you all know this, what's going to happen to the beaker water? Right. It's going to even out, it's going to dissolve. The blue is going to spread evenly throughout the beaker so that, you know, maybe by the time I'm done talking, you might have a fully blue, evenly colored blue beaker, or yellow if you use the IKI. Okay. The blue is a little more noticeable though. So we're moving from high concentration where you just dropped the dye towards the area of low concentration. And the movement is going to continue until it's, you said, evenly blue. The particles are evenly spaced apart. We call this equilibrium. Right? The blue particles are equally spaced within the water molecules. That's equilibrium. I can tell it visibly because I can see it as an even shade of blue. If I gave you a sugar cube instead of the blue or the iodine, and in your beaker of water you dropped the sugar cube, wherever you drop that sugar cube in, that's the area of high concentration of sugar. And over time, the sugar is going to dissolve, and you won't be able to see that there was sugar there anymore. Remember we defined the term solution and colloid and suspension last time? So it's a little bit harder to see with the sugar. I happen to like the blue, but we're looking at the principle of diffusion. Moving molecules from high concentration of those molecules towards an area of low concentration of those molecules. It's passive, it does not require energy. We can move molecules like this through the phospholipid bilayer, just through a process of diffusion called simple diffusion. They just pass right through the spaces in the phospholipids. Or we might need a protein, like we saw earlier. And these are 2 examples of that. We might need a protein in the membrane to make a little channel for us for that substance to move passively from high concentration to low concentration. But all of these examples are passive. They're all moving molecules from high to low without the requirement for ATP.
What is the difference between those two, the channel?
These two?
Mediated and carrier
Mhm.
mediated?
Yep. So with this particular one, we call this, well, they're both facilitated diffusion, because you can't just move the molecule by itself. It's not going to move as easily. And we were hinting to this last time when I said you might need a transport protein to move this medication through the bloodstream, because it's not a water-soluble medication, it's a steroid, something like that, so you need a transport protein. That's essentially what these are. This one, it just stays open. It's in the membrane, stays open, the pore is there from the protein. As long as we have the protein, then that substance can move through. This one will actually change shape depending on what the substance is that's binding to it. So only this substance can bind here and then can pass through into the cell. Whereas this, anything that's this shape or smaller can pass through.
So that the carrier is just very specific.
Mhm. Yep. Yep. Glucose, generally, the way that glucose moves into the cell is like this. There are specific proteins in the membrane that are just for glucose to move in. Will you be able to see any of these proteins physically with your eyes in the lab? No. No. But we'll see the results of them. So if I have my beaker, and you guys do here, with the the blue dye, it's now aggregating towards the top and it's kind of going towards the bottom, right? What could I do to speed this process up?
Stir.
Take a spoon and stir it up, right? What do you do with your coffee when you add the sugar? You stir it, because you don't want to sit there for 20 minutes and wait for the diffusion process to occur. So we could stir it, and stirring it is going to make the molecules move around faster. What else could we do? You ever made simple syrup? If you make like a mixed drink,
Water.
if you know anything about making, not that I do, but you know, if you want sugar to dissolve faster in water, you heat it up. You heat it up. So you're essentially making a simple syrup. So in heating it up is like stirring it. It's causing the molecules to move around faster. And if the molecules are moving around faster, they're going to start hitting each other. You know, we're going to reach equilibrium faster. We could add pressure to it. I could subject it to an electric current. You know, I'm not going to do that, but these are things that are going to make the molecules move faster. They're going to speed up the rate of the process of diffusion, rather than just let it sit there and wait. We could stir it. We could heat it. Increase the pressure. Increase the charge. What about adding more? Can I add more blue to it? If I add more blue or iodine to the beaker and the same amount of water, the molecules are probably going to move around faster. Rather than if I add the water, you know. So I could add more. I could change the concentration of the solution. So with the process of diffusion, we were looking at molecules moving from high concentration to low concentration. And the molecules that you have are methylene blue dye and IKI, iodine. We were only looking at the dye or the iodine in particular molecules. We weren't really focusing on the water. With osmosis, which is a type of diffusion, we will look at the water. We're going to look at whether the water is going to move from high concentration to low concentration. Always water compared to water. High concentration of water, compare it to low concentration of water. And you will be dealing with solutions in both cases. So there will be a salt water solution over here and a salt water solution over here. And you're going to compare the water to the water. Or there could be a sugar solution over here and a sugar solution over here. You're going to compare the water to the water. It's going to be a little bit funny because you'll be thinking about diffusion at the same time. Diffusion of the sugar, which is happening, or the salt, and osmosis of the water, movement of the water. So two different types of movement are going to happen. The sugar might go one way and the water might go another. You with me? Does that make sense? If I have a beaker and I have a 10% glucose solution within my cell, and I put that cell in a beaker with another solution. Maybe this solution has 50% glucose. I could compare the glucose to the glucose and determine which way the glucose is going to move, right? That would be diffusion. Assuming that this membrane is going to be permeable to the glucose, that I would have that special transporter or carrier protein embedded within this membrane of this cell to allow the glucose to move. Which way would glucose move? Right, the glucose will move into the cell because there's a higher concentration of glucose here on the outside. It's going to move from high to low. But you could also think about it with the water. If I have 10% glucose, then I have 90% water. You should not need your calculator for this. Okay, it adds up to 100%. Okay, 90% water now. is inside my cell. How much water is here in the beaker? 50. 50% water. Oh, I abbreviated. And I'm going to compare the water to the water. Now I'm looking at osmosis. Now I'm going to determine the tonicity of the solution. Which way is the water going to move? Good. Water is going to move out. And in general, when water moves out of a cell, it's like you're dehydrating the cell. Water's moving out of it. When you lose a lot of water, what happens? Have you ever sat in the bathtub for a while and you look at your fingers and they look like they're little pruny, like raisins almost? What happened to the cells in your fingers? They lost water. They became dehydrated. They shrunk. They shriveled up. So this cell is essentially going to shrivel because water is going to leave the cell and go into the solution in the beaker. It's going to shrivel up. It's going to lose water. It's going to shrink. We call this solution then a hypertonic solution. The solution in the beaker here is hypertonic. I don't know why my word is shaking all of a sudden.
More water?
Yeah, so hypertonic means when you take a cell and you put it in a hypertonic solution, that cell is going to lose water. The hypertonic solution has less water and more solute than the solution inside of our cell. Yes, we're going to talk about that too. Yes, that's good. Fabulous. Perfect. Definitely, we're getting to that. Does this make sense to everybody? Yes? I don't know if I have enough color or not, but oh yeah, I think I might have another color on this side. So, another example, okay.
Do you give examples of hypertonic in the textbook?
Sure they do. Exactly like mine, no, but yeah, they do. All right, I have 10% sodium chloride in my beaker, and I have, I don't know, 70% sodium chloride in my cell. 10% sodium chloride in the beaker, 70% sodium chloride in the cell. Which way is water going to move? Okay, what's the concentration of water here in the beaker? 90% water. And how about in the cell? 30%. Oh, yeah, no. Water. So I have my high concentration of water here, my low concentration of water here. Water is going to move from high to low. Water is going to move into the cell. So if water moves into the cell is going to swell. It's going to blow up, it's going to bloat. The solution here in the beaker is a hypotonic solution.
The solution in the cell or surrounding?
In the beaker, in its surrounding. When you put a cell in a hypotonic solution, it's going to swell. Water's going to move into it.
So the solution in the beaker is hypertonic?
Correct. Hypotonic.
So when I'm talking about the cell in those terms, it's just what the cell is in.
It's what the cell is in.
Like a cell can't be hypertonic.
It can. And actually, and I wasn't going to talk about this, but we'll always be going from hypotonic to hypertonic. So if we were to just look at the cell itself, that's hypotonic. But here is hypertonic. And water is always going to move from a hypotonic environment to a hypertonic environment. But when I ask the question, I won't ask it of the cell, I'll ask it of the solution the cell's in. you always get hyper, right? You get hyper. So we go from hypo to hyper.
So then do you ever get like the equilibrium?
Yeah. Yep.
Or they just be
Definitely. So if I have a beaker and I have 30% sodium chloride and 30% sodium chloride, what's the water in both cases? 70 and 70, right? 70 on this side, 70 on this side. Which way is water going to move? Both ways. Always will move. And in this case, it's going to move both ways equally. That's equilibrium. This is called an isotonic solution. There's 3 types of tonicity. 3 ways that we can look at the movement of water. Look at osmosis. Either the cell is going to shrivel or shrink, and that's hypertonic. The cell is going to swell, and then the solution would be hypotonic. Or what happens to the size of the cell in this case? It's going to stay the same size. It's going to stay the same size. And that's an isotonic condition. An isotonic does not have to be 50-50 to be even. It could be like I showed you, 30-30 or 80-80. As long as it's even, the same, then it's isotonic. Water's still going to move because it can. There's pores here. It's going to fit through them. Again, all of this is passive. Does not require energy. Does not require ATP to do this. And we're going to test this out. So I'm going to give each group a piece of dialysis tubing. We're going to use that like our plasma membrane of the cell. The tubing has little tiny pores within it, little holes. And smaller substances will be able to pass through those pores. Go from the inside of the cell to the outside, vice versa. Larger substances are going to have a harder time moving and might not move at all. Water will move. I'm going to have each group do maybe a little bit different concentration, and we will kind of collect the results at the end. And you're going to do this at the same time as you will be looking under the microscope, getting more practice with the microscope and focusing in some slides on the cell and hopefully being able to recognize the plasma membrane and the nucleus and the nucleolus and these structures. Questions on these types of tonicity? Okay. So you'll essentially be looking at an experiment like this one. I don't have YouTubes, unfortunately. YouTube, not the video YouTube, but the actual tube that's shaped like a U. It's called YouTube. It actually called that. So with these YouTubes, it's neat because you can put a membrane in the middle. I can put a piece of dialysis tubing right here. And I can put one solution on this side and another solution on this side. And based on whatever the pore size is or the selectivity of the membrane, some molecules will be able to pass and some will not. So the size or the volume of the different sides of the U-tube will change because of that. If I put this solution here on this side, these orange pyramids, then maybe they're salt. Okay, maybe they're starch or some solute. They're large molecules. And the little blue circles are water. So you can see what's happening. The water is moving across to where those solute molecules are. Water is going to always move where it's needed, by the way. Water moves towards the higher area of solute. So if there's more sugar on one side or more salt on one side, water is going to move towards that. If you ever eat something very salty, you eat a lot of potato chips or you eat ham, usually that does it, you get really thirsty. It's this happening within your body. You're trying to dilute the solute, the salt that you ate with more water. So the water is moving across to this side of the U-tube and the volume is increasing on this side. Does that make sense? Yeah? I can't move this solute over here so that they're gonna both be the same. Because the solute will not pass through the pores. The membrane is impermeable, not permeable to that solute.
So hypotonic always equals larger volume, increasing volume.
In the cell, yes. In the cell, yes. And sometimes the cell can stand it, can take the, extra water, and sometimes it cannot. If we were looking at red blood cells, I showed you a red blood cell first slide or so when we were talking about this chapter, and you saw that it had an indent in it where there's no nucleus there. Lost the nucleus, right? So a red blood cell works just like this one here. Just like this one over here. If I change the concentration of the cytoplasm compared to the extracellular fluid, maybe the plasma of your blood, if I put this red blood cell in 100% water, when maybe there's only 99.1% water inside the cell, then what's going to happen to the size of the red blood cell? It's going to swell up. Because homeostasis for the red blood cell is this. This is the cytoplasm. This is what it likes to be. We want our red blood cells to be in an isotonic environment. Right? We want them to have the same concentration, 0.9% sodium chloride on both sides of the cell. So if I have, I already said it, so I might as well write it, 0.9% sodium chloride on the inside of the cell here, and 100% of water here, water is going to move in, the cell's going to swell up. The plasma membrane will not be able to withstand that if we continue with this environment on the outside of the cell. Water is going to continue to move in. It's like blowing up a balloon. You can only blow it up so far before it pops. And a red blood cell will increase in size only to a certain amount before it will burst. Lyse. This is called hemolysis. The red blood cell blows up because you put it in 100% water. If you ever inject 100% water into somebody, that's lethal because of this. The red blood cells will blow up. Same thing over here. So if I have a red blood cell with my 0.9% sodium chloride, 9.1% water, and I place it into a situation which has 1% sodium chloride, what's going to happen? If I have 1% sodium chloride, how much water do I have? I have 99. I have 99.1 over here. Here's my high. Even though it's a 0.1, it's still high. Water's going to leave. The red blood cell is going to shrivel up. It's going to shrink. That's called crenation. When your red blood cells crenate, they dehydrate, they get smaller. Do you think the red blood cell is going to function properly when it crenates? Do you think it's going to function properly when it's going through hemolysis? No. Okay? So it's really important to regulate this and keep the red blood cells in an isotonic environment. The plasma of your blood is essentially the cytoplasm of the red blood cells. Same concentration there. That's what we want. You go to the hospital and you get an injection, you have to get an IV. It's a saline IV. Saline. What's saline? Salt water. Are they going to inject 100% water into you? God, I hope not. They're not going to even do 1%. It's a 0.9% sodium chloride IV. 0.9. Because it's the same, we want it to be the same as your red blood cells, as the solution on the inside of the red blood cells. Because we want them to act normally. We don't want them to get too big or to get too small. It's like three little bears. We want just the right size. All right, questions on that? Yeah.
0.9% of sodium chloride in the water is what makes the water safe for your blood.
Yep, because it's just like the cytoplasm in the inside of the blood cell. And if we change it, if we put too much or too little, the plasma membrane is not going to be the same shape. Maybe the proteins aren't going to function the way that they should. We won't be able to hold on to oxygen and move throughout the blood vessels to transport the oxygen. All these different things are affected. So look at your beakers. Have they reached equilibrium now? The blue ones I know have. I'm not sure about the iodine. Blue, the methylene blue molecules are pretty small, so they'll generally move pretty quickly. You guys put a lot of blue in there, didn't you? I said a drop, not a dropper. Okay. All right, that's fine. So take a 15-minute break. We'll come back and we will look at some cells under the microscope.